Hello, this is Dr. Paningrahi. Welcome to my genetic classes. In this video, I have a talk about the epistasis. So, what is the meaning of epistasis? Epistasis means the stoppage or the inhibiting. Some genes, they have some behavior to mask, to inhibit, to suppress, to stop the expression of other genes. Then such type of behavior is known as the epistasis. In Mendelian experiment, we see that the different allele of the same gene, they interact with each other, and there is a relationship, whether it may be the recessive type of relationship or dominant type of relationship. In Mendelian experiment, out of two, only one allele may expressed. That's why such type of relationship among the allele of the same gene, this is known as the interallelic gene interaction or intragenic gene interaction. The interaction among the alleles of same gene, such type of interaction is known as the intragenic gene interaction. So intragenic gene interaction, the alleles of the same gene, they interact in different way, and which may produce and which may actually behave in different way, and these may be the codominance. Both the allele, if expressed, which is known as the codominance in case of the blood group. The incomplete dominance, as you know, in Mirabilis jalpa or 4 o'clock plant, where the formation of colored flower, this is the one type of example of incomplete dominance. And in Mendelian experiment, as we know, in F1 generation only one character will express, and this character is known as the dominant. So completely single allele expressed in F1 generation of the Mendelian experiment, which is the example of the complete dominance. If the allele of same gene interact with each other, this is known as the interallelic gene interaction. After from different experiment, we also observed that the allele of different gene also interact to each other, and they behave in different way. And such type of behavior, it may be the epistasis, it may be the collaborator gene interaction, or it may be the quantitative gene interaction, or it may be the pleiotropic gene interaction. Here, one thing is common among these four types of behavior of non-allelic gene interaction. So these are the non-allelic, the allele of different gene which interact to each other, and such allele are present in different locus, and such type of interaction is known as the intergenic gene interaction. Alleles of different gene, if interact to each other and produce the new expression of the character, this type of interaction is known as the intergenic gene interaction. It also known as the non-allelic gene interaction. So under such non-allelic gene interaction or the intergenic gene interaction, there is one type of behavior of the gene which is known as the epistasis. So what is the meaning of epistasis? Suppose that this is the one pair of gene which inhibit the expression of another pair of gene, capital BB, which is present in a different locus, distance from the capital AA. So the expression of B, which is inhibited by the presence of capital AA, or capital A inhibit the expression of capital B, or capital A modify the expression of capital B, or capital A stop the expression of capital B, then here the capital A, the gene pair, which is known as the epistatic gene, This is known as the epistatic gene. And the gene pair which is being inhabited, which get inhabited, and the expression of such gene gets stopped, such pair of gene is known as the hypostatic gene. Hypostatic gene. So now we get the complete definition of epistasis. Epistasis, it is the non-allelic or intergenic gene interaction where one pair of gene can modify, can stop, can inhibit, can mask the expression of other gene pair which is present in a different locus. This is known as the epistasis. So epistasis gene if present in recessive form, then it is known as the recessive epistasis. If it is present in dominant form, it is known as the dominance epistasis. And another type of epistasis, this is the duplicate gene interaction. So there are three types of interaction, and I will go one by one serially. So let's start the recessive epistasis. So what is the meaning of recessive epistasis? So recessive epistasis, it indicates that the epistatic gene which present in a recessive manner, if the recessive gene if suppress the expression of another gene which is known as the recessive epistasis, and it is clearly understood by getting of the one famous example, the coat color in mice. So I would like to take the one example about the recessive epistasis. The example 1, the coat color in mice. So the coat color of mice, it is controlled by, suppose that it is controlled by the gene capital C. If the capital C present, then it will produce the black colors in association with recessive small a. If capital C present in association with capital A, then capital C interact with capital A and which produce ego T. If double recessive small cc and small aa present, which produce the albino, color. If small c recessive character if present in association with capital A, then here there is the surprising result that there is no expression of capital A, there is no expression of ego T colors. Here, such individual capital A, when it association with the recessive small c, which actually produce albino. Albino means there is no formation of coloring pigment. So it is obvious that the capital C, it has the ability to express the gene color black, if the capital A in association with capital C if present, then the expression of capital C is modified and which forms the agouti color, if the small c present in association with capital A, then the expression of capital A is inhibited. That's why there is no formation of coloring pigment like black color or the agouti color, which forms the albino. So here it is obvious that the small c gene which inhabit the expression of capital A gene, that's why the small c recessive gene, this is known as the epistatic gene. This is epistatic gene. And the small c which inhabit the capital A gene, the expression of capital A gene, that's why the capital A gene here, the capital A gene here, it is known as the hypostatic gene. Because it gets suppressed, it gets modified, it gets inhibited by the presence of small c. So there is the relationship among the capital A and recessive, the small c recessive, which inhabit the expression of capital A, which is known as the one type of example of recessive epistasis. So agouti color means the hair or the fur which has the alternate black and light, or the hair having the gray color in alternate band of black color. This is known as the agouti color. Black means it is the totally black. So there is one idea. If you observe the total gene position, there will be some precursor molecule. Suppose that there is the precursor molecule A, which is the precursor molecule which convert into the black color, and such black color which may convert into the agouti color. So the conversion of precursor molecule from A to B, suppose that B, which need the presence of one enzyme, and such enzyme actually may regulated by the gene here, capital C. If capital C present, then the precursor molecule A convert into capital B or the black color. And in another step, such black color which convert into agouti color, if there is the presence of another dominant gene, suppose that here in this example, this is the capital A gene. So the capital A gene which may convert the black color or which regulate the enzyme which is responsible for the conversion of black color into agouti color. So here the capital A and capital C both togetherly form the agouti color. So C complement the C which supplement the A, and A also supplement the C. So such type of interaction also known as the supplementary gene interaction. Here the capital C supplement the A, the capital A supplement the C, neither one, there is no ability to expression of equity color. So this type of relationship is known as the supplementary gene interaction. If you cross the two mice having the black colored coat with the albino, then what happened? So this is the one type of supplementary gene interaction. Supplementary gene interaction. If you cross the capital CC and small aa, black mice with the small cc, capital AA, albino mice. This is the parental generation which forms the gamete capital C small a, small c capital A. The F1 generation which will produce capital C small c, capital A small a. So the presence of capital C and capital A which produce the agouti color. As I told earlier that the capital A interact with the capital C and which produce the agouti color. Agouti color. Agouti color means the hair is the gray color having the band of light color. This is known as the agouti color and this is the F1 generation. If you cross such F1 offspring, then what would be the F2 phenotypic expression? So please see, suppose that and the agouti, the F1 generation, which may produce 4 types of gametes. So these 4 types of gametes, it may be capital C, capital A, or capital C, small a, or small c, capital A, or small c, small a. So this is the F1 gametes. So these are the F1 gametes. So I'd like to draw the checkerboard here. This is the F2 generation. The gamete capital A, capital C, the gamete capital C, small a, the gamete small c, capital A, the gamete small c, small a. So capital C, capital A, capital C, small a, small c, capital A, and last one is small c, small a. So in F, this is the F2 generation. So here, capital C, capital CC, capital AA, capital CC, capital A, small a, capital C, small c, capital AA, capital C, small c, capital A, small a, capital CC, capital A, small a, capital CC, small aa, capital C, small c, capital A, small a, then capital C, small c, small aa, here also capital C, small c, capital aa, here capital C, small c, capital A, small a, here also small cc, capital aa, here also small cc, capital A, small a, here also capital C, small c, capital A, small a, here also capital C, small c, and small aa. Here also small cc, capital A, small a, the small cc and capital A. So this is the F2 generation genotypes. So what would be the phenotype? If capital A present in association with capital C, then the capital A the capital C in association with capital A, which produce the agouti color. So it produce the agouti color, capital A and capital C, which produce the agouti color. Capital C, capital A, agouti color, capital C, capital A, agouti color, C and A, agouti color, capital only, capital C, capital A, agouti color. So here also capital C, capital A, agouti color. Here also capital A and capital C, agouti color. Here also capital C, capital A, agouti color. And so how many number of offspring would be produced if having the agouti color? 1, 2, 3, 4, 5, 6, 7, 8, 9. So here 9 are the agouti. And what would be the black color? So those having only capital C, they produce the black color. So here only capital C present, it would be black colored mice. It would be black colored mice, and it also another black colored mice. So here 3 black. So how many number of albino characters are produced? So if small c present in association with capital A, then small c inhabit the capital A, which produce albino. Here also small c recessive, which inhabit the capital A, which produce the albino. Here also small c inhabit the capital A, produce the albino. So how many number of albino mice would be here? So 3 albino. And last one, this is the double recessive, which will be produce albino. There is no dominant. It would produce the albino mice. So here the 1 albino. So here this is albino and this is also albino. So ratio now it would be 9:3:4. So this is the typical ratio of such supplementary gene interaction. And such ratio, and this is the deviation of Mendelian dihybrid cross phenotypic ratio. So Mendelian dihybrid phenotypic ratio is 9:3:3:1. As you know, the 9 means any one capital if present, any both capital. A means any one, maybe here capital. Here also any one, maybe the capital. Here both are the recessive. And here the recessive gene inhabit the dominant gene. That's why the 3 and the double recessive plus 1, which produce the 4. So the 9:3:4, this is the modification of the phenotypic ratio of dihybrid cross of Mendel, 9:3:3:1. So this is known as the supplementary gene interaction, where the recessive gene suppress the dominant gene. Now come to the another example which also elaborately express how the genes actually interact in epistasis condition. So let's have a look another example. This is the example, the flower color, the formation of flower color in sweet pea plant. The formation of flower color in sweet pea plant. So the example 2, flower color in sweet pea plant. The scientific name of sweet pea plant, this is the Lathyrus odoratus, and in sweet pea plant there is the two type of white plants. The some white plants, they have capital CC gene. It is one type of white plant. This is the white plant, white flower bearing plant. And another plant, those having the small cc homozygous recessive but capital PP dominant homozygous present, it also a white bearing plants. So you may say white 1 and white 2. So two types of white plants are present in the sweet pea plant, Lathyrus odoratus, and here you see that any one gene will present in recessive form. In white 1, the P present in recessive form. In white 2 plant, the C present in recessive form. So if in any case, if one should be remain in recessive condition, then another dominant gene will produce the white color. If both the dominant gene capital C and capital P present togetherly, which will produce the colored flower plant. Colored flower means the purple color because of the presence of anthocyanin pigment. So suppose that this is the parent and these are the gametes, capital C, small c, capital P. These are the gametes. And in F1 generation, which will produce capital C, small c, capital P, small p, which will produce the colored flower. This is the F1 offspring. So F1 offspring, this hybrid having the capital C and capital P. So the presence of both the capital gene, both the dominant gene capital C and capital P, which will produce the colored plant. If anyone capital C or capital P is absent, then which will produce the white color. So there is the idea. If you think, then there is the idea, some substance, suppose that A, which convert into the substance or intermediate substance convert into B, and such intermediate substance convert into the anthocyanin pigment, anthocyanin pigment, which is responsible for the production of colored flower. Colored flower. The conversion of substance A to substance B, which need the enzyme, and such enzyme, suppose it is regulated by the presence of gene capital C here for this example, and the intermediate substance B, the conversion of intermediate substance B into the anthocyanin pigment or the color, which need another activity of another enzyme, and such enzyme actually regulated by, suppose that regulated by another capital gene, which is the capital P. So another gene capital P. So here if you observe, if both the genes are present, capital C and capital P, then both the genes togetherly form the anthocyanin pigment. If capital C absent or it will present in recessive form, then there is no chance to the production of intermediate substance capital B. If the capital P absent or present in a recessive form, then there is no chance for the conversion of intermediate substance B into anthocyanin pigment. If both the dominant gene present, capital C and capital P, then the anthocyanin pigment will produce. So it indicates that capital C and capital P, they are complementing to each other. So capital C complement the capital P also complement the capital C. Suppose that. So both the genes complementing to each other, and such type of gene interaction is known as the complementary gene interaction. So you can say this is the complementary gene interaction. Both the capital G, capital C or dominant gene capital P complement to each other for the production of the anthocyanin pigment, which will produce the colored flower in sweet pea plant. So this is known as the complementary gene interaction. And such experiment actually conducted by the Batson. Batson crossed the F1 progeny. Batson crossed the F1 progeny. Such F1 progeny having the capital C and capital P. So obviously it can produce 4 type of gametes. So what are these gametes? Capital C, capital P, capital C, small p, small c, capital P, small c and small p. So these are the gamete of F1 generation. If you cross the F1 offspring, then what would be in F2 generation? What would be the ratio of F2 generation? So please have a look. Let's cross the F1 offspring. So this is the checkerboard. Male and female. The gametes are capital C, capital P, capital C, small p, or small c, capital P, or small c, small p. The gamete of female may be capital C, capital P, capital C, small p, small c, capital P and small c, small p. So what would be the F type? So F2 genotype. So capital CC, capital PP, capital C, capital P, small p, capital C, small c, capital PP, capital C, small c, capital P, small p, capital C, capital C, capital P, small p. It would be capital CC, small pp, capital C, small c, capital P, small p, capital C, small c, small pp, capital C, small c, capital pp, capital C, small c, capital P, small p, small cc, capital P, small p, small cc, capital P, small p. Here also capital C, small c, capital P, small p, capital C, small c, small pp, small cc, capital P, small p, small cc, and small pp. So this is the F2 genotypes. So what would be the phenotype? As you know, if both capital C and capital P present, then which would be the colored flower? If capital C and capital P present, it would be the colored, it would be the colored, it would be the colored. Here also capital C, capital P colored, capital C, capital P colored. There is no capital P. Here also capital C, capital P colored. There is no capital P. Here also capital P and capital C, which will form the colored. Here also colored. And there is no capital C. There is no capital C. There is also capital C, capital P. It would be the colored flower plant. This is, there is no capital P. There is no capital C. There is no anyone capital. So how many numbers of colored plants are produced? 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 number of colored flowered plants produced in F2 generation. So how many numbers of white flower plants? It may be white 1 or white 2, as you say. So if any one recessive, then which will produce the white? So it will produce, there is no capital P, so it will produce white. There is no capital P, it will produce white flower plant. It also produce white. There is no capital C, there is no capital C, there is no capital P, there is no capital C, which will also produce the white. There is no capital C, no capital P. None are present capital, so which will also produce the white. So how many numbers of white flower plants are produced? 1, 2, 3, 4, 5, 6, 7. So 7 plants are white flowered plants. So this is the phenotypic dihybrid cross F2 ratio. But the Mendelian ratio, this is the classical ratio of F2 dihybrid cross phenotypic ratio, 9:3:3:1. 9:3:1. 9 here, anyone, it may dominant, which will produce 9. So here 9 colored, anyone, capital C and capital P, if both dominant gene present, they will produce 9 plants. And if anyone will present in dominant form, these numbers are 3. Anyone, suppose that here capital A present but B are recessive, here both recessive are present. So the ratio 3, 3 and 1 all are produced white flowers. Why? There is no both are dominant. Here only B dominant, A recessive. Here only A dominant, B recessive. Here both are recessive. That's why 3 plus 3 plus 1 is equal to 7. So the Mendelian dihybrid phenotypic F2 ratio, which is modified into 9:7 in case of complementary gene interaction. So this ratio 9:7, which indicates that the allele of different gene interact in the complementary fashion. So I hope you understand such recessive epistasis. If you like, then click on my next video regarding the dominant epistasis. Thank you.